Okay, so at the back of the eyeball, the very back of the eyeball, is a structure called the retina, and the retina sort of covers. It's a series of layered cells of neurons, actually, including some very critical sensory or afferent cells that are going to carry out the stimulus transduction, convert the incoming electromagnetic radiation within a particular wavelength into a neuronal response. And these are called the photoreceptors. So at the very back of the retina, the closest to the vascularization, the back of the eyeball, the most caudal aspect of your eyeball, you're going to find the photoreceptors. And the photoreceptors detect photons, and they're very fascinating and unique. They have this kind of structure like this, and I apologize, this is not complete, this particular photoreceptor, but it has some pretty important components here. It's got this cell body, it has this what they call an inner segment, and then there's the outer segment. And this outer segment is a cone-shaped outer segment. So this is one sort of subtype of photoreceptor cell. This is going to be one of typically 3 distinct subtypes or types of cone cell. And this is what we're going to call an L-type cone photoreceptor, because it's going to be absorbing the longer wavelengths of light, which are going to be, ultimately are typically correlated with your perception of red. But there's like all these membranous discs that are localized back here in this outer segment, and they contain a pigment that will absorb, specific wavelengths of electromagnetic radiation within that range of visible light. And they'll convert that. There'll be a transduction event where it'll be converted into kinetic movement. So the actual pigment will move in response to that absorption. And that will also provoke the release of a chemical that will go off inside the cell, and it will end up closing a channel that was previously keeping this cell actually depolarized below threshold for releasing neurotransmitter, at its synaptic end. So what's kind of odd about the visual system and the photoreceptors in the retina is that they are firing most constantly in the dark before light strikes. Before light strikes, they actually maintain a potential difference, a charge difference across the membrane that's depolarized below threshold, like minus 30 millivolts inside versus the out. And there are voltage-gated calcium channels here at the end, and they'll, let in calcium, and they'll provoke release of neurotransmitter. And so this is called the paradoxical dark current, and it is carried out by the cone cells and also by another photoreceptive cell that's very important in our retinas, called a rod cell. And I have an example of a student made a really nice rod cell here. This has typically a longer rod-like, outer segment with many, many more, membranous discs. So there's significantly more, pigment, photopigment that could absorb, light. And it turns out that these cone cells, there's typically 3 kinds, and they each express their own pigment that can absorb a specific narrower wavelength or set of wavelengths within the range of visible light. But the rod cell expresses rhodopsin, which is the the pigment that'll absorb any visible light spectra at all. So this we're going to see is a much more sensitive cell because it has more surface area for light absorption, more pigment that can actually respond, dance, release the chemical and set off a biochemical cascade and close channels and repolarize, or take above threshold and stop the paradoxical dark current in the light. Well, these cone cells are going to be more selective. We're going to see you need more light, to basically activate them because they have fewer, discs out here, less pigment that can respond to light. You need more light to be around for them to be, to capture light because it's a smaller surface area here. But light absorption for any of these types of photoreceptors at the back of the retina will actually shut off the paradoxical dark current. What's interesting about the paradoxical dark current, like if you close your eyes right now, I want you to know that your cone cells go in like this, your rod cells go in like this, they're firing. They are depolarized, below, the threshold for the opening of voltage-gated calcium channels. So they are releasing neurotransmitter when your eyes are closed. It's the paradoxical dark current. However, that neurotransmitter that they release is actually going to inhibit output from the eye. So you close your eyes, your photoreceptive cells will fire like crazy, and they will inhibit output. They will inhibit the next cell in the network in the retina, and so there'll be no output, really, essentially no output from the optic nerves back to the brain. You open your eyes, and your pupils will dilate the right amount to let in just enough light so that you have some photoreceptors absorbing, and stopping their paradoxical dark current, releasing the inhibition and allowing for output, and others are still basically firing and inhibiting output. So it's a really interesting sensory system and perceptual system with this weird paradoxical dark current. Again, I want to kind of emphasize that a bit because it's as if you think about most sensory neurons, we think about like you touch, for example, and that pressure will initiate an action potential, but actually absorption of light by these photoreceptors will actually shut off release of neurotransmitter, which again will remove inhibition and allow for output from the eye. So the visual system is a series of double negatives, right? So the photoreceptors are in the dark firing. That's the paradoxical dark current, and they're releasing their neurotransmitter onto another cell. This next layer of cells actually has a pole that's receiving the information from the photoreceptors, right, and being inhibited by them when they're firing in the dark, and another pole that goes off and synapses with another cell that's called a ganglion cell. And these cells in the middle here, between the photoreceptors and the ganglion cells, they have two poles, so they're called bipolar cells, and they transfer the activity, either the inhibition or the excitation or the removal of inhibition, from the photoreceptor response, layer to the ganglion cell output layer. And the ganglion cell outputs those axons, that are at the ganglion cells. And it's kind of weird. They're at the very front of the retina. So they're forward. So light goes all the way to the back of the photoreceptors, works its way to the bipolar cells, and then works its way to ganglion cells. And then these cables, the axons from the ganglion cells, will coalesce and form a large cable that'll leave the eyeball and form what we call the optic nerve. And the optic nerve will start heading back, towards the central nervous system. So photoreceptors are found in the retina. They carry out stimulus transduction. They detect photons of light or, light of, electromagnetic radiation within specific wavelength ranges. Because of the pigments that they express in their outer segments back here, right? You have rods and cones back there, and they, oddly and, kind of atypically sort of fire most in the dark when there's no light, when they haven't absorbed any electromagnetic radiation. And that inhibits output, through the bipolar cells and the ganglion cells out the optic nerve, back to the rest of the brain. But when light strikes, well, it'll cause, the pigment here to dance, right? If it's the right electromagnetic spectra, release a chemical, set off a biochemical cascade, close a channel that will actually, kind of repolarize or, take the polarization back above threshold so that this cell stops firing, and that removes inhibition on output, from the eye. So photoreceptors, bipolar cells, ganglion cells, and then just to add complexity, right? Where these photoreceptors synapse with the bipolar cells, with the first pole of the bipolar cell, there are these cells they look kind of like octopi. They're called horizontal cells, and they spread out their connections, and they both receive and send messages, chemical messages. They form synaptic connections with a number of different, cones and rods, etc., of the photoreceptors in a region. And they add some significant complexity to the processing of the signals that are working their way through the networks of the retina. And these are called horizontal cells. They run horizontally at the level of the photoreceptors and the bipolar cells. And then there's another set of cells, and I actually made one here, let's see if I can pull this off without causing a major crisis. Yeah. So this is an amacrine cell, right? This also, like hangs out at the intersection, this time of a bipolar cell and the ganglion cell output, and it will complexify, add complexity to the synaptic connections between the bipolar cells and the ganglion cells, which actually form the output of the eye through the optic nerve. So there's, pretty much 5 cell types. It turns out there's so much complexity in the eye. Your eyeballs are remarkable. Your retinas are extraordinary. There's 20 to, it's been estimated 20 to up to 40 different subtypes of amacrine cell, for example. So there's a lot of different complex, cellular machinery in the retina. It's actually now considered to be a component of the central nervous system. It used to be considered a peripheral structure, but now it's actually considered to be within the CNS. So when you look into somebody's eyes through those pupils, you are looking directly now into their central nervous system.